Hello grade 12s. So I'm going to try and keep this a nice short and sweet video relating to mutations. So just as a reminder, we have gone from the idea of protein synthesis, this important thing for making proteins in our body, and now we have to understand what happens when that process can potentially go wrong. And that's where we're focusing on mutations. Just bear in mind before we start with this, don't think that all mutations are necessarily bad. They are not all bad. Mutations are happening all the time, and some of them are actually good. We need mutations in our life, but we have to understand how the mutation works if we are to understand how it's going to affect an organism. So, just as a reminder of what we've done so far, this is normal protein synthesis, what we want to occur. So there's a DNA strand with a set sequence of nucleotides. I'm then going to make a complementary mRNA strand, and then I will make my protein based on the set amino acids that I've been reading off based on the different codons that I have here. But let's say, for example, on the DNA strand, I take this cytosine, I remove it, and I replace it with a thymine. What is that going to do to my mRNA strand? And what is that going to do to my protein? What if I took this nucleotide away completely? How would that affect my mRNA and my protein? Well, that's what we're going to find out about with regards to mutations. But first of all, let's give ourselves a definition. What do I mean by mutation? So a mutation is a permanent change in the DNA of an organism. So something that is not going to revert back. It has happened. It's not going to change back. And specifically in the DNA of an organism, in the recipes, the instructions telling us how to make the proteins. Now, importantly, this may, not necessarily always, but it may result in a change in function or create dysfunction. Again, it could do nothing, could be harmless, but it may result in these changes. So, there are two types of mutations that we can group together generally. So the first is grouped as point mutations, and these are referring to changes in a single nucleotide. So it's only affecting one single nucleotide in the whole DNA chain of nucleotides. The second type is a chromosomal mutation, and that is where you have a change in an entire chromosome itself. So the whole chromosome is affected at once. We are going to focus on point mutations in this video. When we do meiosis, we'll discuss chromosomal mutations. So, what can cause these mutations? Well, the first, as part of the natural process of DNA replication and all these kind of things, it can happen by random chance, spontaneously, through errors with the enzymes, the polymerases, and all those helicases, all that kind of stuff. It can cause an error to occur. The DNA is folding in on itself. It can create a wrinkle where there wasn't supposed to be. Nucleotides fall off and attach on where they shouldn't. It can happen. The other way that it can occur is through mutagens. So mutagens are substances that if you are exposed to them, the more exposure you get, the greater the chance or the likelihood that you are going to cause a mutation. They literally cause changes in the DNA itself. So examples of these are things like X-rays, radiation, UV light, and chemicals. So any prolonged exposure to these can increase your chances of getting a mutation to occur. So that's why, like when you go to hospital, you try not to have too many X-rays at once. Don't go out in the sun too often with the UV light, gives you skin cancer, mutation. Asbestos as part of chemicals, giving the lung problems. So that's really where the 2 points of mutation can come from. So, with regards to point mutations, there are 3 types of point mutations. I'll go more in depth into them as we go, but very quickly, this is our original sequence of DNA. So the first type is an insertion deletion. So as the name suggests, I am inserting, I am adding in an extra nucleotide where there wasn't one before. So notice that it has this word frameshift here. That's going to be important because everything that comes after this insertion is essentially shifting along further down in an insertion one. We'll discuss that a bit more just now. Second type is a deletion mutation. And as the name suggests, I am removing one nucleotide. Where it was in the strand here, it is no longer here. This is also a frameshift mutation. Everything is now shifting towards where the deletion occurred. And the last type is a substitution. So I originally had thymine on this DNA strand, and now it has changed to cytosine. This is not a frameshift mutation. Everything after this change has stayed the same. There is no shifting this way or shifting that way. This has remained the same. Okay, so those are your three. Let's look at them a bit more closely. This is an example of an insertion deletion. So up at the top here, I have my original strand of DNA, where these are my 5 codons. Remember, a codon, a group of 3 nucleotides, and the 5 amino acids that they were coding for. But now what has happened is right here, I have inserted an extra uracil. So essentially, this is on the mRNA that has happened. So on the DNA, I have had an extra adenine that was added on the DNA, which meant I have an extra uracil on the mRNA, because it's now a permanent change. It's always going to cause that. Do we now notice that even just in this codon, I have changed the amino acid that I am making? I was making tire, now I am making lu. But now what's the problem? If we look after the mutation, I have also changed this amino acid that I was coding for, and now I have this lonely guanine that's sitting here, but it can't code for anything because I need to have 3 together. And anyway, it's 1 extra than it should be. So now I have changed the amino acid sequence. That essentially is an insertion mutation. Deletion works the other way. So now I've removed this uracil that was, so I had the C that was here, and now I have shifted it along. So now this is gone. So there was a guanine on the DNA. So the guanine is gone. So now I have shifted along. So now in the original, I was coding for 6 amino acids. But now, in this particular example, I have gone 3 amino acids and then the stop codon, as in stop making the protein. But I've still got some other codons and these 2 lonely adenines that are sitting here. So now I've completely changed what is happening in my protein. It could be that you don't always necessarily have a stop codon. This is just an example of what I've got from here. But it will, again, everything after the deletion will change the amino acids that I'm creating. So the insertion and the deletion are frameshift mutations, where everything after the insertion or deletion has shifted either one away or one towards, which therefore changes all the amino acids that come after that. Then we get to a substitution mutation. So in my original, the only one that has been affected is this codon here, where I originally coded GAA, which gave me glue, but now I have changed the 3rd base of the 2nd codon to cytosine. So now I've got GAC, which is making ASP. So but then everything after this change has stayed the same. So it's only this one amino acid that has changed. So this is not a frameshift mutation. This is just a normal point. I've only changed the one amino acid. So now I've had all these changes. But what does that mean? So what is the effect? Well, you can have one of 3 things is going to occur. Because of the change in the protein, it is either going to be beneficial to the organism, neutral, or harmful. So beneficial being that it is going to benefit, it's going to be of an advantage to the organism. It could be something that is like a phenotype change, a different color coating that allows you to blend into your background more, something along those lines. Harmful, obviously, is then going to be deleterious to the organism. It could be a dysfunction, something could not work, it could make you more vulnerable to prey, something along those lines, whatever is harmful. Neutral is where there is no change. So even though you've had a mutation, it is not going to have an effect on the organism. Let's just have a look and quickly see how that's going to work. So remember your little codon table over here. Remember when you were looking in each of the blocks, you may have noticed that sometimes, let's take this block over here, even if I changed the third base of this codon, it doesn't actually matter because I'm still going to code for the same amino acid. So I could have CUU, CUC, CUA, CUG on my mRNA sequence, and I will still be making LU. Same way I've got like over here, I'm always going to make pro as long as the first two bases are the same. So this is where you could have a neutral mutation. I'm still having a substitution, or it could be a deletion or however kind of thing, whatever it is, as long as I'm still coding for the same one. Generally, this only happens in a substitution where you will get a neutral. If you have a frame shift, it really is going to affect the whole protein and therefore the organism in some way. How do I know whether it's going to be beneficial or harmful? Well, for your cases, you will always be told if this is beneficial or harmful, because it will normally be in a case study format. There is no way for you to know that if I suddenly changed all the amino acids after a certain sequence, whether it's going to be good or harmful for the organism. It will have to be told to you. The one you will have to figure out for yourself is if you have this case where you are told it has changed the third base, but it is still coding for the same amino acid. Therefore, it is definitely going to be neutral, which is therefore something that you're going to know. The only other time that you could potentially know something is if, in the example like I gave you with the deletion, if halfway through a protein, it suddenly starts coding for stop. Then it is a fair bet to say that this is going to be a harmful mutation, because essentially you are not creating a full protein anymore. So as opposed to it being a different protein in some other way, you are now creating no protein. So you could almost automatically assume that that is going to be a harmful mutation to that organism. But we'll do some applied examples in another video for us to go through that. So as a link towards evolution for later when we do the evolution section, mutations are important. Mutations are allowing us to have variety, which allows us to work with the different environments and adapt. Adaptation is only able to occur through mutation. So when we get into actual evolution section much later, this will start to make a lot more sense as to where it becomes important to us. Now, some of you might be saying, well, how often does this mutation happen? If you're saying it happens all the time, surely we should all be messed up in some way, shape, or form. Remember, we only deal with the basic stuff. So for your purposes, this is what we deal with. But do understand that DNA is very clever. It still has its own checks and balances. It has lots of enzymes that are inside it to make sure that it is actually not making mistakes. It can go and look through a whole section of DNA and it can know this is not supposed to be here. This is in addition. This wasn't supposed to be here. So it does have fail-safes to help it deal with these insertions, deletions, all those kind of things with the frame shift. Substitution's a little bit more difficult, but it can still pick them up. When those start to go wrong is when you really start to have the disorders happening. So for example, cancer, which is the uncontrolled cell division, is really where the mechanisms stopping your cell from doing too much dividing are not in check. And that mutation has gone far beyond what your body is able to control. So I hope that helps you in terms of understanding mutations. Go back, pause, rewind, make sure you make notes, have a good understanding, and I really hope this video did help you guys out. I'll be doing some applied things in a video coming soon to go through some questions, and then you'll be able to check your understanding from there. Otherwise, thanks very much for joining me. Skype handle is always there if you need to get me. Teams, get a meeting. Otherwise, happy studying and enjoy your time, guys.